Well, it's a real pleasure to be here. I thank Siri very much for the invitation. And finally, I was able to accept and be here. It's been a few years in the making. There's a little bit of a history to me standing here. Um, it really is a pleasure to be here. And today I'm going to talk about something that is not necessarily the bread and butter of my lab, but it's a project brought to me by a postdoctoral fellow in my lab. And it turned into be a really interesting project. So that is what I'm going to show you today. Hopefully I can work this. Okay. So as we're thinking of CF, we're obviously all striving to get to the cure. And that, for many people, we think about gene replacement, gene correction, or even cell therapy, which is kind of what we've been interested in looking at, the feasibility of a cell therapy becoming a reality and what that would take. So when we're thinking of regenerative medicine approaches, we need to think about things like what cell are we targeting? Does this even matter which cell we're targeting? How many cells do we need to target? Can replacement cells functionally engraft and persist in the lung? For how long? And how do stem cells behave in a diseased environment? And it's that last point that I'm kind of going to hone in on. We think about the epithelium and how the epithelium is functioning, what it's doing and performing its barrier, but that epithelium receives signals from the environment that it sits in. And the stem cell, which is the main cell that regenerates an airway epithelium when it becomes injured, receives cues and signals from the environment that it sits in. And it's often forgotten about when we're studying disease in a dish. So we all know that CF is associated with inflammation, and inflammation has a significant impact on airway function. In fact, in CF, abnormal airway inflammation is present very early within the first few weeks of life, even in the absence of infection. And this progresses over the years of life and remains abnormal throughout the life of someone with CF. People with CF are obviously susceptible to repeated and chronic infections, leading to elevated airway inflammation. And this inflammation results in a significant change in that stem cell niche. And this is just an image here. Pointer. This is an image here of an airway in a CF lung. The airway is full of mucus. We can see that in this area here, we've got what we call basal cell hyperplasia. Basal cells are the main stem cells in the airway epithelium. We've got excessive mucus in the submucosal glands, and we've got huge infiltration of inflammatory cells. It not only happens in the lung, it happens in other tissues impacted by CF. Here is an example from wrong button. I'll get the hang of this in a second. This is an example from a pig model of cystic fibrosis looking at the pancreas. And again, you can see a huge amount of immune cell infiltration associated with the exocrine pancreatic insufficiency and pancreatitis, which occur in cystic fibrosis. These processes of inflammation and pancreatitis are also commonly linked to pancreatic cancer. And as people with CF are now aging, we're seeing an increase in the incidence of cancers and other things that the lung dysfunction used to kind of overshadow. So inflammation is not only just related to the pancreas and the lungs. Many of the other organs affected in CF, such as the GI tract and kidney, also are impaired by inflammatory processes. So now we're in this post-modulator era. We've seen obviously a significant impact on the quality of life of patients with CF. Generally, we're getting improved FEV1s, overall improvement in lung function. We've seen a reduction in the exacerbations, improved quality of life, well-being, and significant improvements in life expectancy. However, there are still challenges remain. It's not the end of the road. We've still got things that need to be worked on. And there are patients who still get chronic infections. Inflammation seems to persist in the lung and other tissues. Some patients have chronic and ongoing GI challenges. The incidence of CF-associated diseases, so the CF-related diabetes, liver disease, kidney disease, is increasing as the population with CF is now aging, and now we're seeing more complications in this area. Obesity is actually something that is becoming more prevalent among the CF population. CF patients, I'm sure you're all aware, of the lack of nutritional value, so they've been taught to eat more calories. And now with the modulators, that is a challenge that it's difficult to now change, and we're seeing an increased incidence in obesity. The reason I've highlighted these here is that a lot of these complications are associated with inflammation. So my talk today, I've split it into 3 parts, and it's not necessarily in the order that I want to tell you it in, but it's in the order of what I believe is the most important order. We're going to start with, I am not getting the hang of this thing. Start with the impact of cystic fibrosis transmembrane regulator on macrophage function. So macrophages is one of the key immune cells in the body. We're then going to move on to how does a CF epithelium talk differently to immune cells. And then the final part is developing more physiological or pathophysiological models using human cells in a dish. So this first part, as I mentioned earlier, was really a project brought into the lab by postdoc Ben Calvert. And he was really interested in looking at how immune cells interact with the airway epithelium and how there's crosstalk between the two and how the epithelium signals to the immune cells. And his fellowships kept getting rejected. And one of the biggest criticisms, and it was a constructive criticism, well taken, is that each donor is different. So when we're looking at primary patient cells, we have significant donor-to-donor heterogeneity. So interpreting that in a dish is incredibly challenging to find out what is significant and what is related to CF when you've got such donor variability between your controls and your CFs. So he went away and thought about how he could tackle this, and this is the story that evolved. So we know that CF macrophages are different. We know that they're hyperinflammatory, and there's been research done in the past that has shown that there's increased macrophage numbers in the airways during fetal development. And this correlates with elevated levels of monocytic chemoattractants in the bronchiolar alveolar lavage fluids. We also see that patients have elevated interleukins, interleukin 4 and 13, in response to Pseudomonas infection. This suggests that macrophages in CF are perhaps tolerant to these interleukins, which these interleukins typically will promote a phenotype of a macrophage that is anti-inflammatory, or an M2 macrophage. These are not as prevalent in CF patients. We also know from a number of studies that CF macrophages tend to be suboptimal as phagocytes. So phagocytosis and key phagocytic markers such as CD11b are suppressed in CF phagocytes. Markers that would be typical on your M2 macrophages, the anti-inflammatory macrophages, are also suppressed in CF. And there's been studies showing that acidification of the phagolysosome, which is critical for killing the bacteria, are decreased in CF. Is decreased in CF. So we know that there's an impact on phagocytosis. And here's just a number of papers that have tried to show that this is related to the cystic fibrosis transmembrane regulator. However, it has been very challenging to really definitively state the role of the CFTR in these processes, largely due to the cellular heterogeneity and the donor variability, and really defining that this is a CFTR-related process. And this was a nice a figure from a nice paper from Ben Kopp's lab at Emory that showed that when you have a non-CF macrophage and treat it with Cepacia here, you can see that CFTR, which is in green, is on the cell membrane of your macrophage, and you get functional bacterial killing, where you can see the red bacteria inside the cell is not so bright, not coming through so great on the screen. But when you have a CF macrophage here and infected with Cepacia, you can see that the bacteria persist and there's no CF expression here. And here they're showing that if you treat it with some of the modulators, that you can see an improvement in CFTR expression and some level of bacterial killing. Though you can probably appreciate there's still a significant amount of bacteria present here. So it's not fully restoring phagocytosis. So this is where the challenges are. Really confirming CFTR expression and its precise function in tissue-resident alveolar macrophages in the lung has been really challenging due to these reasons that I've mentioned: the cellular heterogeneity, the donor variability, and the capacity that macrophages have is their ability to rapidly adapt to changes in their environment. So when you take them out of the body and manipulate them in a dish, they've already changed. So are you looking at the CF phenotype anymore? So it poses many challenges when trying to really define what is going on with macrophages in CF. So this was Ben's solution. He went away and thought about how he could do this in a controlled way. And he decided to take advantage of this THP1 monocyte human cell line. This cell type can be stimulated with PMA to differentiate the monocytes into macrophages in a controlled manner. What he decided to do was capitalize on the gene editing technology using the CRISPR-Cas9, where he could go into these normal THP1 cells and actually insert a delta F508 mutation. So we did the gene editing, we clonally expanded these cells and generated a pure line of cells that are now genetically identical to the parent, except for they contain a delta F508 mutation. We also created CFTR knockouts and have a line of those too. He then did basic characterization of both the delta F508 mutants and the wild types and showed that their phenotype was very similar in response to PMA. He could differentiate both lines into macrophages. And when he looked at the cell surface profile, which included these markers and a larger panel, there was no difference in the phenotype of his monocytes and macrophages. But then the question is, are they functionally different? Does having a delta F508 mutation in your macrophages change their function? And he does this by stimulating them with a lipopolysaccharide to stimulate the inflammatory response. And what he saw was that there are 3 cytokines here. So we're looking at interleukin-1 beta, TNF alpha, and interleukin-2. The red is the wild types, the blue is your CF. The left column of both bars is the vehicle control, and the right column is the LPS stimulated. And what you can see is that in the F508 mutants, there is a significant increase in the interleukin-1 beta, in the TNF alpha, and in interleukin-2. And interestingly, there is a significant decrease in the anti-inflammatory interleukin-10, all suggesting a pro-inflammatory phenotype has been induced in these cells by adding the delta F5 mutation. We also looked at phagocytosis. We did this using a pH Rhodo assay, so this fluoresces red upon acidification, and we monitored this over time. And again, the wild types are always in red, the CFs are in blue. What you can see is that there is impaired phagocytosis in the CF macrophages compared to the wild types over time. This is summarized in the graph here, and we use cytokeratin D here to maximize this process. And again, on the right-hand side, this is just a visualization of it. We have our delta F508s on the top and our controls on the bottom. You can see the red signals we're getting from the acidification of the pH Rhodo. But what I hope you can appreciate here is the significant bacteria, which is kind of the grainy background you can see here, compared to the bacteria quantity that is in our wild types. So you can see a significant increase in the existing bacteria, indicating a failure of phagocytosis in the delta F508 mutants. We also collaborated with Cheng Yong's lab at Oklahoma State University. He's been working with these cell lines as well, and he did a bacterial killing assay for us using Staphylococcus aureus. And again, you can see here over 24 hours and at 72 hours, there is a significant impairment in the bacterial killing due to the delta F5 mutation in these cells. So Ben then wanted to ask, can we restore this using the modulator therapies? Are the macrophages responsive to modulators, and can we restore phagocytosis and reduce the pro-inflammatory phenotype? And as I mentioned earlier, this paper here suggested that yes, some of these modulators can do this. So Ben went in and started looking at Trikafta. So he used elexacaftor, ivacaftor, and tezacaftor in combination, and he looked at our inflammatory cytokine production. And again, wild types are red, CF is the delta F508 mutants are in blue. You can see that actually in both wild types and in the CF, there is a reduction in interleukin-1 beta production in response to a dose-dependent increase in the ETI drugs, almost bringing the CF back down to the levels of the wild types. So we thought this is great, they're responsive. But then we looked at TNF alpha, and the story was a little bit different here. What you can see in the wild types with increasing doses, at the lower doses, not much happens, but when you get up to the higher doses, which we believe are still in the clinical range here, you're seeing a significant increase. In fact, this is a huge increase in TNF alpha production. And the same happens in the CF. Well, there's a very little effect at the lower doses and at a higher dose. So in a dose-dependent manner, we're seeing an increase in pro-inflammatory TNF production. So then Ben wanted to ask the question, is this due to a specific modulator in the triple therapy, or is it due to the combination of all three being given together? So he broke it down and looked at each modulator individually. And we're looking at the same cytokines here, interleukin-1 beta on the left, TNF alpha on the right. And again, interleukin-1 beta, we seem to see some reduction here. However, the TNF alpha production in both the wild types and the delta F508s is increasing in response to ivacaftor. Moving on to tezacaftor, we see very little change in response of interleukin-1 beta to tezacaftor treatment. However, similarly, the TNF alpha is increasing in a dose-dependent manner in both the wild types and the CFs. And then finally, we looked at elexacaftor. And again, in the interleukin-1 beta really isn't changing, but there are some slightly smaller changes in TNF alpha in response to elexacaftor. So it seems like all the modulators in some way are impacting TNF alpha production in these macrophages. So looking at the individual drugs on phagocytosis, again, we've got time across the bottom axis and your concentration of bacteria on the y-axis here. Red is your wild types, and blue is your CFs, and shades of it are different doses, which are highlighted on the right, which I appreciate are probably very difficult for you to see out there. But bottom of the story is that there is no change in phagocytosis in response to the 3 drugs that we tested here at any of the doses that we gave. When we put them in combination, so it's combining the ETI together, we do see that there is some suppression of phagocytosis. So rather than an improvement that we would expect to see, there is actually suppression at the higher doses in phagocytosis. And this also happens in the wild type as well as the CF. So this is not specific to CFTR dysfunction, but the higher doses of the modulators are leading to a decreased phagocytosis. So Ben then went in and started to profile the macrophages and see is there anything changing in the cell surface profile of these macrophages. And he did a huge panel that I'm not going to show you of cell surface markers indicative of the different types of macrophages. And this is just after 1 hour of exposure to the modulator. So this is a very quick response, not time to get transcription translation changes here. But what he sees is that CD14 on the cell surface is significantly increasing after 1 hour in response to the modulators in both the wild type and the delta F508. CD14 on the cell surface is pro-inflammatory and is also tied to TNF alpha production and regulation. So we've kind of seen a little bit of a cycle here that maybe there is something going on that is leading to a change in the pro-inflammatory status of these cells that is independent of CFTR. He then has, this is actually very recent data, and it doesn't look as clean because he's pulled a lot of experiments together here, but you can still appreciate that elexacaftor seems to be the drug that is driving this upregulation of CD14. If you look at ivacaftor and tezacaftor, there's very little difference, but elexacaftor or the drugs in combination seems to be what is driving this. So I will come to what Ben's hypothesis is here. This is not a closed book story. It's still an investigation that we're continuing with. But what we see is that CFTR does directly influence the inflammatory and functional responses of macrophages. I think we've shown that now where everything is the same, but that delta F508 mutation. We've generated paired wild type and delta F508 THP1 cells that can be a very useful model for looking at macrophage function in people with CF. We've shown that ETI combination therapy is not necessarily improving the function of macrophages, and that higher ETI combinations are actually driving this pro-inflammatory cytokine release and suppressing pathogen clearance. And that elexacaftor seems to be the one that is contributing to potentially trafficking other proteins to the cell membrane, including CD14, as we just showed you. So this has led to Ben's hypothesis right now that elexacaftor is actually driving the trafficking of CD14 to the macrophage's surface. And that CFTR is actually, his hypothesis is that CFTR expression is actually critical on the phagal lysosome and not on the macrophage cell surface. There's data out there showing that its expression on the phagal lysosome is regulating the acidification, which is then critical for bacterial killing. And what he thinks is happening in response to modulators, and modulators are doing their job. They're trafficking CFTR to the cell membrane and away from the phagal lysosome. So he's thinking that the modulators may be having an adverse effect on bacterial killing through this impact on CFTR expression in the phagal lysosome. And that we're also getting this concurrent trafficking of CD14 to the cell surface, which is increasing TLR4-induced inflammation regulated by TNF alpha. So he's going to keep working on this to prove the mechanism here, and hopefully that will give us some potential targets that may help to reduce inflammation in patients that are on modulators with sustained inflammation. So that was data that I thought was incredibly exciting and potentially quite important. But all this project came around because of Ben's desire to really understand how the epithelium and the macrophages interact and talk to each other. So that is what I'm going to move on to tell you about. So just to remind you that this is the CF niche, and this is a cross-section of an airway. We've got the airway, which really is not looking very healthy on the apical surface here. And you can see all of these green cells underlying. These are all neutrophils. So this is not typical of an airway. This is typical of a CF airway where we have neutrophilic inflammation changing this microenvironment. And in addition to the inflammatory cells that are in this microenvironment, we also have extracellular matrix, and we also have other cells, other neighboring cells. And often when we create a cellular model in a dish, we look at the epithelium, or we look at a macrophage, we look at extracellular matrix. We don't necessarily put them all in combination and think of this as a tissue level model. And that is one of the challenges of research. The more complex the model gets, the more hard it is to interpret it. But it often means that we're overlooking some specific phenotypes in our cells and things that we're looking at. So Ben took these traditional air-liquid interface culture models that we use to study our airway epithelium, and he's done a few things with them. He's worked on creating different types of integrated co-culture models using macrophages. And then we've evolved these to emulate biosciences, microfluidic chips, and developed an airway epithelium model on this, which we published a couple of years ago now, the original model, but we're now using it to study macrophage epithelial interactions. So this was some of the earliest work that Ben did. He did co-cultures in submerged culture, where you can see green macrophages with red epithelial, airway epithelial basal stem cells here. This is called his submerged model. And then he has the air-liquid interface lifted culture and a fully differentiated airway epithelium. We have three model systems, one where the macrophages are integrated within the epithelium. This has been the most challenging and still is a challenge. One where the macrophages are in contact with the epithelium on the basolateral side of the membrane. So there is some contact here. And then ones where the macrophages are on the basement membrane, on the base of the well underneath and are not in direct contact. So whatever they're secreting or changing can be interacting with the epithelium. And we can use each of these models to ask different questions. So these graphs are a little bit complicated, and I apologize that I haven't broken down, but on the left, the green and the red bars, which I realize I apologize to anyone who's colorblind. The left is the green, and then we have red. These are co-culture models. And then the right-hand bars, which are blue and gray over here, these are monocultures. So by monocultures, I mean that we have the epithelium cells alone down here. These are the macrophages alone, and these are epithelial cells with macrophages. To complicate this, we have different combinations of CF and wild type. So we have CF epithelial cells with wild type macrophages, CF with CF, and vice versa, wild type epithelium, CF epithelium. So you can get where I'm going here. There's a lot of combinations. And when you're doing this in a donor to donor way, you've got to decide which donors you're pairing with which donors. So to eliminate some of the complications, he used Newly and Kufi cells, which are cell lines from wild type and CF. just to eliminate some of that donor heterogeneity, so he could really look at how the macrophages were interacting with the epithelium. So these macrophages were isolated from donors and grown in the co-culture systems using this underside model. And Ben's interested in the inflammatory response, so he started by looking at changes in the inflammatory response by taking washes from the apical side and the basolateral side. Here we're just seeing the response for TNF-alpha production again. And the one thing I want you to take home from all this data that's shown here is that monocultures of macrophages, so these are wild types, these are CF, monocultures of macrophages are releasing TNF-alpha. When you put them in co-cultures with the epithelium, we see a suppression of that TNF-alpha release, and there's the same quantity of macrophages in each of these assays. So there is an interaction with the epithelium that is actually helping to regulate the TNF-alpha that is secreted by macrophages. So Ben looked at other cytokines, and it wasn't always the same situation. Here is looking at interleukin-6 production, so another pro-inflammatory cytokine. You can see that in epithelium alone, there's not really any release. In macrophages alone, there is some release, not much difference between the wild types and the CFs. But when you put these in the co-cultures, you see that there is a substantial increase in interleukin-6 production. And if you look down here, there is actually a significant increase between the wild types and the CFs. And the CFs so this is the CF with the CF macrophages. This is the CF with the wild type macrophages. And then these are wild type with CF, wild type with wild type and wild type with CF. But the take home is that co-cultures of CF with CF have increased interleukin-6. And this is when you stimulate them with LPS, you can see that this is significantly elevated in the CF-CF co-cultures. So again, suggesting that there is some interaction between the epithelium and the macrophages, leading to changes in pro-inflammatory cytokine release. And finally, he saw the same very similar effect with interleukin-8, and I hope you can appreciate that these numbers are very large. So where you're seeing this significant increase here in the CF models, we can see that there's much more interleukin-8 being released in co-cultures with CF epithelium and CF macrophages. And some of that is coming from the CF epithelium alone, where you can see that the CF epithelium here without stimulation is already increased at the tonal level without having LPS stimulation. So interleukin-8 is key because interleukin-8 is one of the main attractants for neutrophils. If we've got changes in interleukin-8, the likelihood is that you're attracting neutrophils into the vicinity of the epithelium. So this increase in interleukin-8 in these co-cultures could identify a potential target to help prevent airway neutrophilia. What he also showed was that signals from the, and this title, oops, sorry, wrong button. This title's a little misleading, actually. So this is the statement that actually we need to read. So what he did here, we haven't done this assay yet with the CF epithelium, but we took wild type, so non-CF bronchial epithelial cells, and cultured those with non-CF macrophages. These are primary macrophages here, CF macrophages, or just use the the non-CF macrophages. I have to apologize, got this the wrong way around. It's non-CF epithelium with CF epithelium, and these are all non-CF macrophages here. So we simplified it by sticking with the non-CF macrophages. And what we can see is that when you have the non-CF macrophages with the non-CF epithelium, we can see that there is more phagocytosis happening. When you put the non-CF, so the wild-type macrophages with the CF epithelium, there is a suppression in phagocytosis. This suggests that the CF epithelium is signaling back to the macrophages and impacting the macrophage function, and not typically as you would think about it the other way around, where the macrophage functions responding to the infection and influencing the epithelium. So this kind of proves Ben's hypothesis that the epithelium and the CF epithelium is a key driver in changing immune cell responses in the CF epithelium. He also showed here, and this is something that we're particularly interested as a lab in ciliogenesis and mucociliary clearance, but he also showed here that when you grow the macrophages in direct contact with the epithelium on the underside, so you can see here the outline of a macrophage that is grown on the underside, with the CF epithelium, with the epithelium as it differentiates. So this is comparing non-CF macrophages to CF patient-derived macrophages. This is taken after 28 days of air liquid interface culture, was typically sufficient to mature and polarize the air epithelium. And typically you will see these nice cyan cells, which are ciliated cells, and this nice tight junctions, which are marked in red of the epithelium. What you can see here when you've got CF macrophages with the exact same donor wild-type epithelial cells, there is a substantial impairment in multiciliary genesis at this time point. Again, suggesting that signals here from the CF macrophages are regulating airway stem cell function. Now, I do have to say we did follow this out for much longer and took it to 3 months. And by 3 months, we did see that the ciliogenesis had been restored. So it seems to be something that can be overcome over time, but there is an initial impact and impairment on differentiation of basal stem cells in the airways and the presence of CF macrophages compared to non-CF. And the other thing Ben has observed is that when we look at CF macrophages, again, compared to non-CF with a wild-type epithelium here, we can see that these CF macrophages are naturally hypermigratory. So over time, you can see that these ones on the left here are actually remaining on the underside. You can see it's nice and flat and laid out on the underside. There's another one up here in the CF macrophages. You can see that many of these highlighted by the red arrows have migrated to and up and through the epithelium and are now appearing on the apical surface. So it suggests there are functional differences of CF macrophages when they're cultured in the environment of a wild-type epithelium here. So we're wanting to pursue this and keep moving on and looking at all the different combinations, but hopefully those THP1 mutant cells that we've now generated will actually aid us in being able to interpret this and eliminate some of the donor variability and make it so that we can actually interpret the data in a statistically relevant way. So I've got one, I'm not going to give you the summary right now. I want to move on and go through the final part. So this is work that was really led by Jana Nauwirth. She has recently left my lab and is now running her own lab at the Helmholtz Munich in Germany. But she was really interested in moving these models into tissue-level models. And she brought this technology into my lab. This is a chip from Emulate Biosciences, where we have two channels. We have an air interface channel in blue and a blood interface channel in red. And these chips have the potential to provide more of a tissue level model because you can have the epithelium, you can have air interface, you can have stretch, you can have physiological function in there. You can have a blood interface lined with endothelial cells where you can put immune cells through. And you can do real-time analysis of these chips as processes, when you want to evaluate a process. So when we brought this into the lab, I thought, this is great. These are going to be awesome. They're going to transform what we're doing. And 2 years later, we're still working out how to culture the cells on these things. But we finally did get it working. And the paper was published in collaboration and with huge help from Anne van der Doos, who works in the Netherlands using the same chips for airway. We were able to eventually get the cells to grow and differentiate. And we see very little differences in the phenotype of the cells from a standard air liquid interface chip, transwall culture to the chips. But this was the biggest problem we had to overcome, was that these chips that are stretchable, so to allow us to have the function and put stretch on them, the membranes have to be more flexible, and this means having larger pores in them. Well, the larger pores, epithelial cells love to migrate through, and what we were seeing was the cells migrate through and reside on both sides of the membrane and don't differentiate and make a pretty useless model. But after a long time, we found that treatments with an anti-adhesive coating actually was able to prevent this from happening long enough that the cells would polarize and differentiate. So then we did a lot of characterization of these models to make sure that we could really model airway function on them. And we looked at ciliated cell function. So these graphs on the left are really showing that there's not a huge difference in ciliated beat frequency, so the function of the cells. There was a difference in the area of the chip that was covered with multiciliated cells. We saw that the chips after 14 days had significantly more differentiated and functional ciliated cells than the inserts did. We looked at mucociliary clearance. So again, this is like data you saw yesterday. We're tracking bead particles, and we can see that the chips actually after 14 days have great coverage, but also great directional flow of the cilia compared to inserts at 14 days where you see very few ciliated cells and very little directional flow. This dynamic flow actually is really key in maturing the airway epithelium too. And what we saw was that in the chips, markers of planar cell polarity, such as Vangal, you can see that you're starting to get significant organization and polarization of these cells with expression of Vangal on the outside edges of the cells, whereas in the inserts, these have not yet organized themselves and developed planar cell polarity. This is something that was significantly increased in the chips. We'll just skip over those, they're not particular data. The exciting thing that what we thought this was exciting was that, we looked at the epithelial cell types, and although we don't see huge differences between transwells, what we do think is that the chips are actually leading us to a more smaller airway phenotype, which would actually match the level of stretch we were giving to the airways and the flow dynamics we were giving. So we think that these may be able to help us model different branching generations of the airways, and it's something we're looking into. But what was interesting is when we add airflow in these, we have directional airflow, the cilia actually orient themselves and beat against the airflow. So you can see here, this is without airflow. You can see that there's a tendency to go towards the inlet over here, but there's no directional beating. When you add airflow, you get very organized directional beating. So we believe that these models are actually a really great way to study airway function. We did look at the impact of airflow and stretch on extracellular matrix gene expression, and we saw some significant differences to transwells where you're seeing a reduction in some of these matrix proteins and matrix metalloproteases. And we just validated that at different levels. And the reason I highlighted these is that these are some genes and matrix components that are actually significantly associated with airway remodeling that occurs in CF and other airway diseases. So the fact that these are different in a functional chip to an air-liquid interface culture may have important implications for studying their role in disease. So these are my final couple of slides. We've now got to the point where we culture in our CF patient cells on these chips. We trust them enough that we're not going to waste our precious CF patient cells. And we started to look to see if there are any differences between the CF cells and how they behave when we have airflow and stretch on these chips. And we've now got an N of 3. I don't have statistics on it, but this is something that we found quite interesting, is that our wild-type chips, we get very distinct mucociliary clearance in a directional manner. And you can see that cilia motion is even across the culture. When you look at the CF chips, we see that although there's good quality beating, the beating is not necessarily perfectly aligned, and there seems to be some dyskinesis in how the cilia are functioning. And I thought this may be just some donor specific response, but we've now repeated this 3 times, and it's holding true that we're seeing some dyskinesis when you have airflow and stretch. So it may be indicating a new component to dysregulated clearance in CF airways that we've not seen when we've had a contained transwall culture. And another thing that we noticed in these cultures, we added neutrophils to try and mimic our airway neutrophilia here and compared. This is a wild type line on the top and a CF line on the bottom. Again, this is a representative example. We've now done this in 3 independent donors, seeing similar responses in them all. The red is the neutrophilic marker, and you can see after 30 minutes of culturing these in the basolateral blood channel, with just the CF epithelium present, you can see that the neutrophils are already migrating up to the epithelium, and after 90 minutes, there's a substantial amount of them come through the epithelium, actually appearing on the apical surface. We see 0 of them coming through on the wild types. And just to prove there are neutrophils in the wild types, these are the neutrophils remaining in the basolateral channel. So again, the CF epithelium here is talking differently than a wild type epithelium to your inflammatory cells. So to summarize the talk, I think what we've seen here is that there is crosstalk between the epithelium and immune cells, and this can significantly alter the tissue level inflammatory responses. We've seen that airflow and stretch can alter the properties of the epithelium and the function of the epithelium. And we've shown that CF macrophages are functionally impaired due to CFTR mutations. and that modulators actually seem to be having an increase in pro-inflammatory cytokine production and further decrease in phagocytosis at higher doses in both non-CF and CF macrophages, indicating that maybe in a patient-specific manner or a donor-specific manner, that ETI may be confounding inflammation and be part of the reason why we're not seeing resolution of inflammation in all patients. And that this seems to be due to a mechanism that is maybe driven by Lexicaftor trafficking CD14 to the cell surface and also trafficking CFTR to the cell surface of macrophages, as opposed to the phagolysosome changing bacterial killing properties. So hopefully I've given you a little bit of food for thought here. It is not a polished story yet. We're still following up on these investigations, but this work was largely led by Ben Calvert, my postdoc, Jana, who I said has already got her own lab in Germany now, and Shuba, Eric, and Greg are some of the other key players in these specific projects. So thank you all for your time.